Hello and welcome to GeneLab Chats. Today we have the privilege of interviewing Fiona Samson from Troy High School. It's known that space travel often has detrimental effects on organisms, like accelerated aging in humans, and researchers work to mitigate those effects. Fiona and her teammates conducted transcriptomics analyses of a fruit fly dataset, GLDS207, from the NASA Open Science Data Repository, and they proposed that targeting a specific protein could help slow the cellular aging mechanisms. In this interview, Fiona describes how the GeneLab data systems and the GeneLab for High School Summer Training Program contributed to this publication. Thanks for joining us today, Fiona. Could you please highlight the most important finding of your study?
Hi, thank you for having me today. As accelerated aging is a very prevalent problem when it comes to spaceflight and is detrimental to long-term space missions, in our study, we focused on addressing this problem and identified a gene of interest called RDGA that could potentially influence aging in spaceflight. Analysis of RNA sequence data from Drosophila that was sent to the ISS indicated that spaceflight significantly affected gene expression in the flies. Spaceflight specifically decreased the expression of the retinal degeneration A gene, or RDGA as I mentioned earlier, in mutant flies that survived the 30-day space mission. And this gene encodes for a protein called dicycloglycerol kinase, or DGK, which modulates the activation of the mechanistic target of rapamycin, or the mTOR, signaling pathway. And that pathway is known to regulate aging negatively. Therefore, DGK may be a potential target for promoting longevity under spaceflight conditions, which could eventually lead to breakthroughs when it comes to advancing long-term spaceflight.
Thanks for sharing that highlight with us. My second question is, how did the GeneLab data systems or the GeneLab for High Schools program help assist you in your work so you could achieve these research goals?
Yeah, of course. So the GeneLab data system contained all the data repository that we accessed to jumpstart our study on the fruit flies. And we utilized the RNA sequence data that was in the GLDS 207 file for our study for our data analysis. And we took advantage of the many statistical analysis tools on the platform. And the GeneLabs for High School internship program provided us with the necessary training and guidance that we needed to use these tools to the best of our ability. And this internship connected me with my team, who I conducted this research alongside with, and also provided us with mentors who provided us much guidance with the overall design of the study and assisted us in presenting our findings at the American Society for Gravitational and Space Research Conference, and then eventually also helped us publish the research in our research article in the Gravitational and Space Research Journal.
Thanks for sharing that with us, and I'm glad to hear about your positive experiences. And we appreciate the time you spent to share this information with us and our viewers and to our audience out there. You can learn more about this work, explore resources from the GeneLab for High Schools program, and access the datasets analyzed in this work with the links provided in the description section below. Thank you and have a wonderful day.